We looked in our section on matter at different types of biomolecules that are important to living organisms. Some of the ones we looked at were carbohydrates, we looked at glucose, we looked at triglycerides, which are a lipid. We also looked at proteins, which are made of amino acids. We looked at a molecule called ATP. And we also looked at a nucleic acid. We looked at one specific type of nucleic acid called DNA, or deoxyribonucleic acid. And for about the past 60 years, we've been learning a lot about DNA just in different fields of science. Biochemistry has helped a lot. Chemists have helped. Biologists have helped. Geneticists have helped. Okay, but the first two scientists to really introduce this idea of the shape of DNA were Watson and Crick, and that was in 1953. And they introduced their idea that DNA was in the shape of a double helix, or almost like a twisted ladder. Okay, so we knew DNA was the substance of inheritance. Some work had been done by a guy named Morgan. We'll talk more about him later on how DNA was inherited. Okay, but we knew that information was encoded by DNA and it reproduced in all cells of the body. And that DNA and the information encoded in the DNA directs the development of biochemical, anatomical, physiological, and a little bit of the behavioral traits of organisms. So there's Watson and Crick with their model of DNA. Whenever they were, I can't imagine knowing the pieces and being able to fit it together in such a way that it makes sense and it's perfect. So we'll talk about why it's perfect and how we figured out how it works and how it's put together. All right, so I mentioned Morgan. His group showed that genes are located on chromosomes. So genes are little sections of chromosomes, and there's components of chromosomes, the DNA and the protein, and each of those were a candidate for genetic material. So we didn't really know which piece was contributing the genetic information, but we knew they were both there. Okay, so the role of the DNA in heredity was first discovered by looking at bacteria and viruses that infect them. And the discovery of the genetic role of DNA happened in 1928 with a scientist named Frederick Griffith. And Griffith worked with two strains of bacterium. One was pathogenic, meaning it caused disease, and the other one was harmless. So he looked at the results of infections from each of these bacteria after he manipulated them a little bit to figure out that DNA was the genetic material. Okay, so what Griffith did was he mixed remains of the pathogenic strain, and so he boiled it, heated it somehow, and he mixed the remains of that heated pathogenic strain with living cells of the harmless strain. And what he figured out was that when he did that, those living cells who had been harmless became pathogenic. Okay, and so what he did was transform the harmless bacteria to contain DNA from the pathogenic strain, causing that bacteria to produce certain proteins that caused it to be pathogenic. Okay, so he called it transformation, and it was a change in the genotype and the phenotype due to this uptake of some new DNA. And so what we're looking at here is kind of a cartoon drawing of Griffith's experiment. And so he took living S cells, okay, that was his control, and living R cells, just to see what would happen when the bacteria was alive. With the S cells, okay, that killed his experimental organism. It killed his mouse. Okay, the R cells, which were the harmless bacteria, didn't do anything when he injected them into the mouse. Okay, so if he killed the S cells, which when they were living killed the mouse, okay, nothing happened. Okay, he had used heat, he had killed the S cells, didn't bother the mouse after they were killed. But whenever he mixed the heat-killed S cells, which were normally harmless by themselves, with living R cells, which were also harmless by themselves, the mouse died. Okay, so Griffith was not really sure what was going on, but what he figured out was that these living cells that had been harmless were taking up something from the heat-killed cells. Okay, so about 15 years later, a scientist by the name of Oswald Avery and his group, announced that they had figured out that the substance that was doing the transforming from Griffith's experiments was DNA. Okay, and it was based on more experimental evidence that only DNA worked in transforming the bacteria into pathogenic. Many biologists were still skeptical, just because there was so little known about DNA at the time. Okay, so these are bacteriophages, and these are viruses that infect bacterial cells. And so what we see is this phage head on the virus. We've got a tail and fibers that connect to the bacterial cell, and we've got this DNA that's being injected into the bacterial cell. And so that's how bacteriophages are able to infect bacterial cells. Okay, so Hershey and Chase, not quite 10 years after Avery's work, performed, I like to think of them as the blender experiments, and we'll look later at why they're called blender experiments. But what they figured out was that it was the DNA that was being injected, not the protein that was being injected into the bacterial cells. Okay, so the way they did that was they took these bacterial cells and the bacteriophages, and they dyed the protein of the virus with one dye, a sulfur dye, and they dyed the DNA of in the bacteriophage with a phosphorus dye. Okay, so both of these are radioactive. They can view the dye and see where it's at and if it's moving from the virus to the bacteria. Okay, and so what they did, they took all these cells and they got them mixed up, and what they found was that the only radioactive dye that made it into the bacterial cell was the phosphorus dye. Okay, which had to mean, because the DNA was the one dyed with the phosphorus, that was what the bacteriophages were injecting into the cell. If it was the protein that was the hereditary material for the virus, then they would have found radioactive sulfur in the bacterial cells after they blended them all up, centrifusion, which spun them around really fast and pelleted out cells. They would have found that dye, the phosphorus or the sulfur dye, in the pellet if it was protein. But because they found the phosphorus dye in the pellet, they knew that the DNA had to be the molecule from the virus that was being injected into the bacterial cells. Okay, so we know, and we've looked at this, and at the time they knew that DNA was a polymer of nucleotides. Each one had a nitrogen base, a sugar, and a phosphate group. And then in 1950, Erwin Chargaff came along, and he figured out that the composition of the DNA, so the A's, T's, G's, and C's, were in different amounts for each species. Okay, and so that kind of lended a little credibility to the idea that this DNA was coding for genetic material, because it was variable with different species. And so that's what we would expect if we have variation within species. We would expect our genetic material, whatever it was, to be varied also. Okay, and so we have now, thanks to Chargaff, these two rules whenever we're building DNA. Okay, we know that the base composition of DNA varies. Okay, but Chargaff's second rule is that in any species, the number of A and T are equal, and the number of G and C are equal. And so when Chargaff measured that, it kind of clicked when he's thinking about all this that there must be an A lined up with every T, a G with every C. Okay, so prior to that, and prior to Watson and Crick, the idea was that adenine bonded with adenine, cytosine with cytosine, guanine with guanine. But what we got, if you put a model together like that, was this kind of wavy, not uniform ladder. And so it didn't really make sense. But then Chargaff figured out that the amount of A is equal to the amount of T, G is equal to the amount of C, and so they must be the ones that are bonding together. Okay, so we've got our structure there, our phosphate and our sugar, phosphate, sugar, down the backbone, and then our nitrogen bases here are T, A, C, and G. Okay, so once DNA was finally accepted as genetic material, now we just had to figure out how its structure helped it carry out its role in heredity. Two scientists by the names of Maurice Wilkins and Rosalind Franklin used X-ray crystallography to look at the molecular structure. And so Franklin produced this picture, and it's now a famous picture, of a DNA molecule using this X-ray crystallography. There's Rosalind with her X-ray diffraction photograph. And so by looking at this photograph, Franklin figured out the shape of a DNA molecule. Okay, and so that allowed Watson to figure out that DNA was helical, and the X-ray images also helped Watson to figure out that the width of the helix and the spacing was pretty uniform, and that the pattern in the photo helped lend more support that the molecule is made up of two strands, which formed a double helix. So there's our double helix. If you flatten it out, you've got the 5' end on one side, the 3' end, with our backbone kind of flipped, and we end up with these different models of our DNA structure. Okay, Watson and Crick were able to build models of a double helix that lined up with the X-ray crystallography and the chemistry of the DNA. Franklin concluded that there were outer sugar phosphate backbones with nitrogen bases in the middle. I remember Chargaff helped come up with those rules for how those bases pair up. Okay, and Watson built a model where the backbones were anti-parallel and the subunits ran in opposite directions. Okay, first, like I said, Watson and Crick thought that A bonded with A, C to C, G to G, and T to T. But like I said, the pairings didn't produce this uniform width in the helix. And so what they figured out was that if you put a purine, which is one type of base, and a pyrimidine, which is another type of base, and you bond those together, you always get this uniform width, which is what Franklin saw in her X-ray data. Okay, so if we bonded a purine with a purine, we got this helix that was too wide. A pyrimidine and pyrimidine, it was too skinny. But if we had a purine with a pyrimidine, it matched perfectly what we saw in the X-ray data. Okay, so Watson and Crick figured out that the pairing had to be more specific. Adenine paired only with thymine, which matched Chargaff's rule. Guanine paired only with cytosine. And Watson and Crick's model does explain Chargaff's rule that the amount of A in any species matches the amount of T, and the amount of G matches the amount of C. Okay, and there's our bases. Got our adenine and guanine that bond pyrimidines, thymine, and cytosine.